Hi everybody, Dr. Brazier here, and I would like to welcome you to Lab 6, Seedless Non-Vascular Plants. Now, the purpose of this lab is to actually introduce you to the fundamental components of plant evolution. And we're going to start with the basic, most primitive land plants. Now, I wanted you to get a good idea of what they look like. So as you watch this video, you'll notice these flat, kind of glossy plants, and those are liverworts, which we're going to look at. And then you'll see this blanket of green kind of grow behind them, and that's moss. Now, to let you know, this time lapse covers 4 months. And I just think these are spectacular, amazing organisms, beautiful, and very unique. You can see this liverwort. What makes it so unique is you can see those little cups on it. Those are called gemmae, and they're really awesome because if you look inside them, you can see these little green things growing in there. And essentially what they do is allow this plant to reproduce asexually, which means that it can just reproduce new growth without having to mate with another plant organism. Just really interesting stuff. Probably makes me a bit of a nerd, but I'm okay with that. So essentially what we're looking at here, we're going to begin with these most primitive land plants, the seedless non-vascular plants. And we are going to work up to the most advanced and dominant form of plant life that we always think of when we think of plants are the angiosperm. Now, to let you know, you can see here are the very simplistic, more primitive land plants. Probably shouldn't say simplistic, just different and more primitive. Now, you can see that this is a cladogram or a very kind of simplified tree that's showing the evolution of plants from the simplest plant to the most advanced plant. And you can see that you have this bifurcation that happens. We've already kind of talked about these and you've learned a little bit about how these work. And so at each one of these points, it's saying that there was an evolutionary event that allowed a change to create this kind of liver, like in this case, a liverwort versus, what came over here and diverged and created mosses and this other section of organisms. And so what this tells us is that this green algae is essentially the ancestral plant to ancestral organism to plants. And we know green algae is essentially found bathed in water. So when we talk about plants, we need to talk about how in the world did they develop into an organism that is able to flourish and survive on land versus where their ancestors are characteristically found in water. Now, these guys can be divided into domain eukarya, and if you remember, eukarya means that eu meaning true, karya meaning nucleus, and so they have nuclei, which helps to protect their DNA, and they have membrane-bound organelles, and we learned about this in Bio 1. So if you can't remember this, you can go and look at the basics of the cell and be specific. You want to look at plant cells and not animal cells because they are a little bit different. In the supergroup, it's the Archaeoplastida, and the kingdom is Plantae. And so in order for them to flourish, they needed the following characteristics. First, they needed to figure out a way that they could prevent water loss. They were in an environment that was water rich before. And so in this case, they developed what's called a waxy cuticle, and there's this protective waxy layer to keep things in. They also developed vascular tissue, and this helped to support and transport nutrients and water around the plant before they could use the basics of diffusion or osmosis to get things to where it needed to go. And then the last is the presence of an apical meristeme. Now, these meristemes give rise to the primary plant body and are responsible for the extension of the roots and the shoots. And so this area is capable of division and growth in these root and shoot tips in plants. So that's why this is important. So these are the 3 main characteristics that plants now exhibit to allow it to flourish and survive on land. Now, what we're going to be focusing on this week is seedless non-vascular plants, which means, yes, they do not have seeds, and they are non-vascular, which means they have what's called a thallus. They don't have the typical xylem and phloem that what we considered vascular tissue for plants. But they do have a means to allow material to get around the plant, which is the thallus. Now here, these are called the amphibians of the plant kingdom. And the reason why is because they need to be in a very moist, water-rich environment. They're not going to be submerged in water all the time as if they were in a lake or a pond, but they do need water to allow to facilitate the means of reproduction. Now, these are known as bryophytes, and typically there are 3 phylum that are within this particular subsection of plants. It's the phylum Hepatocophyta, there's the phylum Anthoceratophyta, and phylum Bryophyta. And so we are going to focus on an example of each. If you look at the Hepatocophyta, is liverwort, and we are going to look at a specific type of liverwort in this lab that's called Marchanta. Now, the Anthoceratophyta is typically an example of this is hornwort. We will not be looking at this in this lab. And the Bryophyta is moss, and that's the other organism that we're going to be looking at. So there's 2 main organisms that we're going to be focusing on in this lab. I'm going to start, now when you get to the lab itself, it's going to first start with the liverwort and then go into moss. I start with the moss because I feel like it's a little bit more simplistic in the term that it doesn't have both, when we're looking at this, asexual and sexual reproduction that we're going to focus on. We're focusing on sexual reproduction here. And so you can see in our example that we have both a male and a female version of this moss. And these 3 are examples of the different stages that you see within the formation of gametes and spores for the female moss and the male moss here. Now you can see that they have what are called rhizomes in the bottom, and these are root-like hairs. And then you have the leaf structures here that you can see in both of them. And then in the male, we're looking at this area here, which is the antheridium. And so here's where we're going to find the gametes that we know. This is the male sex organ, right, that produces sperm. Now, over here, if you look, you'll notice that the female is a little bit more complex because, of course, they have the gametophytes, and these gametophytes here, we looked at the one in male, and then you can see that this is the gametophyte that is for female, right? And so these are the gamete-producing structures. This is what's going to produce the eggs and the sperm. And then you have the sporophyte, which you see in the female, and that is the spore-producing structure. It originates, as you can see right here, from the gametophyte. And the difference between these 2, you'll look at them, is that these are typically diploid in nature, which means they're going to have 2N, which means 2 sets of chromosome, 1 from each parent. And then the gametophytes are essentially going to be haploid, or 1N, because you're going to have the female DNA or you're going to have the male DNA. Now, this top area that you see is called the capitulum. And what originates or comes out of that is what's called a seta, and this is the stalk. Now, at the end of the stalk, you have the capsule, and this, you can see the capsule right here. Now, the capsule can get a protective kind of covering over called the calyptra, which is this structure that's here. Now, the operculum is this little thing that's right here, and it basically covers and holds over the nucleus, which is the opening or that round ring, until the spores are ready to be released. And these are the spores that are within the capsule. And so I wanted to show pictures over here, and as you go through, you can use these cartoons is what we call them. I hate that they're called cartoons because, I have an idea of being a child and, watching the Smurfs or whatever that is. And that's my idea of a cartoon. And I remember being in one of my biology classes and chemistry both, and them saying this cartoon of this. And I'm like, this is the worst cartoon ever. So, but yes, you can use these images here to help you to identify, hopefully, when you look at the pictures in the lab. And so here, right on this bottom, is the male, and you can see this kind of brown structure that's here is what you're looking at, that antheridium that is here. And that this up here is the female, and you can see this whole thing right here is the gametophyte. You can see the gametophyte here in the top section that's right there, which is a capitulum. And then you can see the seta coming out and the essentially capsule, and then the cover that's around it, and you can see it kind of come up there is that calyptra. Now, when these first get solidified and start to grow, there are these thread-like structures that start coming out which are called prototinium or protonium. So we're going to look at that too. But this is the overall to help you with the anatomy of the plant. Now this is essentially, we have in plants what is called alternation of generations. And lab plants have this kind of sporadic life cycle, which means they make spores. And it involves 2 generations that alter between each other. So basically, during alternative generation, the plant alternates between a diploid stage called a sporophyte that actually produces spores, or plants that produce spores, and a haploid stage called a gametophyte, which means gametes that are gamete-producing plants. Although all plants go through alternation of generation, one of the phases, either a gametophyte or sporophyte, dominates in a given life cycle of every single plant group. And so essentially what happens here is, I'm going to start with this down here because I think it's the easiest part, is the gametes. And what are gametes? And we can think about eggs or sperm, whether, in whatever we're looking at. And basically they come together to be fertilized and they become what's called a zygote. Now, that zygote will allow it to produce what's called a sporophyte. And at this point, because we have the gametes coming together into 2, right, that 2N, that sporophyte is now considered a diploid. So everything on this is considered diploid. So it will produce a sporangium, which is essentially allowing the formation of a spore, right? And then this guy will divide in half based on meiosis. And if you remember, meiosis was talked about in essentially somewhere in the middle of bio 1. So this is when you have a 2N, and through 2 cell or nuclear divisions, or cell divisions too, you get an N cell. So you have meiosis 1, and if you remember, you have prophase, prometaphase, metaphase, anaphase, telophase. And essentially, to start with, you have, in humans, like the double number of chromosomes. And then from there, it divides into half and then divides again. So you end up having half of the number of chromosomes that you need. And so in here, it's that same way. And then the spores essentially have 1N. And they get released and they grow and they become gametophytes. And then those gametophytes make gametes and the whole cycle goes around. So anything on this side is haploid, anything on this side is diploid. And as we go through this, you'll notice and you'll want to remember all of this, like what side is what side. Okay, so now what we're going to look at is, we saw that just generalized, simplified diagram of the alternation of generations. So what I thought I would do is, let's first watch the video to look at what it looks like underneath a microscope and all these different parts on a moss. And then we'll actually go into a cartoon that represents it as well. So it's starting to grow. And that's the protonium. Those are those kind of lines that you're seeing there. They're showing all the different gametophytes. Now these are the hair-like structures, the rhizomes, towards the bottom. that's the seta coming up. That's part of the sporophyte, and you can see the cap there, and you can see that kind of ring or circle around it. And over time, that's going to remove and allow the spores to come out. You can see them flowing out there. They're very small. They're numerous as well. So within these plants, every plant has a dominant stage. And for moss, it's the only, or these bryophytes, they are the only group of land plants that have a dominant gametophyte. So let's kind of take a look at this life cycle and how it works. And so if we remember, you can see here, we have both the male and the female. And you can see that the rhizomes are down here, this hair-like root structure, and then you have these non-vascular stems that are coming up here in the leaves. And then they're looking at these caps. And at this point, these guys right here are haploid. Remember, only one set of chromosomes, and that should make sense because they're both male and then independently female. Now, if you look, here is the male. And you can see the antheridium that are over here that actually hold the sperm. And essentially, they'll release those, and it can flow into water and come into this archegonial head. And you can see here, these little things that come out here are venters, and within those, it'll go into that venter and go down and fertilize that egg cell. And so they're showing you up here what it looks like, right? And so here is the venter. Here is the neck of that. And essentially what happens is the sperm comes down here, fertilizes that, right? I'm just gonna put and then it becomes a zygote. So at this point, we're at 2N. And that is going to start reproducing by mitosis, right? To make a bigger and bigger set of cells there, which is called an early embryo. Now, over time, what that's going to do is you can see that these ventral walls are starting to enlarge, and it's going to produce out of there from that. Here, down here's the foot, and you can see this comes up, and the seta is here, and then it's going to produce the capsule. And at the early stage, it's going to be mostly capsule, and then you'll start getting the developing calyptra. And then over time, you're going to get this whole sporophyte here. Now, what happens with this is you can see at the end here, the seta is here, the capsule is here. The calyptra leaves. And you see the operculum is here, and that opening allows all those all those spores to release. At those point, because you've had meiosis happen, we're back into 1N. And so that's going to move, and you can see the spore case that's right here. And this is a process called meiospores, or spores that are being produced. And you can see it starts producing the rhizomes, the little head, those little hairs that are there. And then you get the proteinium, which are those thread-like structures. And then you'll start getting buds that come off of those, and those become gametophytes. And then from here, They'll develop female, male, and then the process starts over. So that's a typical life cycle. So as you go through the lab for moss, and I'll show you, they are going to look at different pictures, and they're going to ask you to draw, say, the gametophytes and the sporophyte. And then they're going to ask you to identify the different male and female sex organs and identify where gametes are located and ask you if things are haploid or diploid. And they're going to look at the sporophyte and have you be able to label things like the stalk and the capsule and the seta and the operculum and the spores in general, and ask you general questions about all of those, as in what are they, are they haploid, diploid, what are dominant stages, all of those things. Now, the first thing that you're going to cover in the lab, we're going to talk about the liverwort life cycle. I feel this one is just a tad bit more complicated, which is why I saved it for discussion second. Now, we already know what some of these structures are called. So you can see the rhizoids there on the bottom of this leafy-like structure here called the thallus. This is what we have in lieu of the vascular tissue like xylem and phloem. And so here are hair-like structures. This thallus is going to originate and have this kind of stalk that comes out that produces on a female an archegonial head and antheridial head on a male gametophyte. So these are the actual gametophytes for liverwort. Now, what will happen is on the archegonial head, you can see the archegonium is right there. And within this archegonium is where the egg is. Now, the antheridium here holds the sperm, and the sperm will be released here, can flow through air, sometimes water, and it can go into the actual archegonium and fertilize the egg. Once the egg is fertilized, know that sperm and egg are 1N. These gametes are haploids. And then this will be a 2N or a diploid at this point, the embryo. And then from there, you will get the seta that will start growing on this foot area. And then here are the spores, and you'll get a capsule and all that. And then the spores will release. And then essentially, just like in moss, it will start growing the rhizoids, and it will basically go through the same processes. Now, what makes these things a little bit different is if you look on the thallus, they have what's called a gemma cup as well. And essentially within the gemma cup, there's what's known as gemma. And this is for asexual reproduction. They can just release gemma. It can go to a different location area and grow another liverwort. So these can produce both sexually and asexually. Just remember, they do have the sporophyte that you can see at the top and the gametophyte at the bottom. And they have the same idea as it comes through this simplified alternation of generations. And so let's watch the video. This is going to be an animation for it versus I just loved that moss video. I thought it was really nice. But it'll go through. The sound isn't the best. I apologize, but I thought it was a really good video nonetheless.
The common liverwort, Marchantia polymorpha, has two alternating stages in its life cycle. The gametophyte and the sporophyte. It is dioecious. That is, it has separate male and female gametophytes. The male gametophyte produces stalked antheridiophores, which bear antheridia on the upper surface of their tips. The female gametophyte produces stalked, umbrella-shaped archegonophores, which bear archegonia on their undersurface. When conditions are wet, the flagellated sperm cells swim from the antheridia to the archegonia, which contain the egg cells. They then fertilize the eggs, thereby producing diploid zygotes. The zygotes divide mitotically and eventually develop into sporophytes. Each sporophyte remains attached to a gametophyte. Haploid spores are produced in the sporangia by mitosis and then released. They soon germinate and begin dividing mitotically to form gametophytes. Liverworts can also reproduce asexually, vegetatively, either by fragmentation of the thallus or by means of gemmi. Gemmi are groups of cells that are produced in gemmi cups on the surface of the thallus. They float away when the cups fill with water and then develop into a new plant.
Isn't that just epic? It's so cool. I just can't believe that. It's really neat. And so just to kind of go through this as a review on what is happening, we'll start up here where we have the kind of mature plants. And so we have a male here, and that leafy-like structure is the thallus. And then you can see the female over here, and you can tell the female is the female because the little gyanophore is like an umbrella shape. The antheridophores are kind of more like a tree, kind of bushy out. So that's how you can identify the difference between male and female here. You can identify the difference between male and female in moss if you look at kind of like the cap that's on the gametophyte and if something has actually produced the stalk or not, right? And so on here, what will happen is the archegonium has the egg again, and the antheridium has the sperm, and the sperm gets released and will go up that channel. Fertilization happens, and at this point, all of these are N, and at this point, it becomes 2N with the zygote. That will start dividing. You can see there's a ton of different cells in there because it starts dividing by mitosis here. That is when things get bigger and grow. And so essentially over time, this is going to generate the foot area here and the seta, which will be the stalk. And then you can see this capsule that's here. And this whole area right there is the sporophyte, and these are the gametophytes that are here. Now, this capsule will essentially then release at some point, and the spores get produced after mitosis. So at this, you have 2N, but when you have the spores actually produced, the spores will become N. And so just like in us, if we go to make a new egg or make a new sperm, we have to split half of the essential chromosomes in half. So that's what's happening here. And they disperse, and then they will go into soil. They'll germinate, and they'll start producing those rhizoids that will lead to the thallus and then produce this new plant. Now, just a reminder, they do not have a true stem because they lack these vascular structures. So now let's look at our pre-lab questions for this week, and hopefully they will seem very intuitive and you will not have a problem answering those, right? So sorry about this. Plants arose from what type of ancestor? And hopefully you remembered it was green algae. And now let's talk about what domain, supergroup, and kingdom do they belong to? Eukarya, if you remember, true nucleus, right? Archaeoplastid is the supergroup. And I remember plastia, because essentially plastids are these things that hold or store things like a plastid. There is a type that you find in regular plants that's green, and that's where chlorophyll is. There are types that hold starch in plants, and plants are red and for instance, a red bell pepper because the chromoplast. So that's how I remember this. So if you went through Bio 1 and you remember looking at identify chloroplast, chromoplast, a myeloplast, which is what potatoes have with starch, then you'll think of, okay, they're within the plastids because they have these plastids. And then kingdom, Plantae. I always feel like when you're studying and when you're looking at this, try to reference it to something that you already know. Because if you can reference it to something you already know, then you're more likely to remember. It's kind of like when I meet someone for the first time. I try to think, okay, if they tell me their name, who is it that I know that has that name so that I can try to make an association. Like there's a guy that lives across from me, and he basically has the same first name as my boyfriend's best friend's wife. And because it's a gender neutral name. And so I remembered it because I was like, okay, she's the only person I know with this name. That'll help me remember his name. So doing those kind of things in all aspects of life, not just in your everyday, but in classes like this can really help. So what are the characteristics of plants that allow them to move on land? And so remember, we had the waxy cuticle. And if you remember, that helped with water loss. You had vascular tissue, which helped with support moving nutrients and water around. And the presence of the apical meristem, which is the whole precursor to primary plant body and extension of the root and shoot so that it could stay in the ground and then get the nutrients from it. So define alternation of generation. And basically this is just another word for sporic life cycle. And so this is just involving generations that alter between each other. And so in this, we have the gametophytes and the sporophytes. It's just going back and forth. If a plant is dipole, which is 2N, is it a sporophyte or is it a gametophyte? And if it is 2N, then we know it is a sporophyte. Are spores haploids or diploids? And hopefully you remember that I have different questions here. Oh, sorry, that goes here and that goes there. I missed it. The gametophyte always gives rise to gametes. Is a gametophyte haploid or diploid? And so we know here this is haploid. And this is the dominant of the two. So that's the first set of pre-lab questions. And then we have the second set of pre-lab questions because the first set came after introduction to plants in general. And then the second set of pre-labs come after introducing seedless non-vascular plants. Do seedless non-vascular plants exhibit alterations of generation? And hopefully you know, yes, because that's what we've been doing, right? What is the dominant generation in seedless non-vascular plants? And we know it is the gametophytes. During alteration of generation, the gametophytes produce haploid gametes, and the sporophytes produce spores. Hopefully you know they are haploid through the process of meiosis. Name 3 phyla of seedless non-vascular plants. Hepaticophyta, which you remember, hopefully, that this is what we get liverwort. Enterocertophyta, and the bryophyta, which is our moss. So what are the two that we're looking at today? Liverwort and the moss. And the liverwort type that we're doing is Marchantia. Why are these plants popularly called amphibians of the water kingdom? And basically it's because they are located close to water and use it to reproduce. We need water for that, the sperm to be able to flow into the female reproductive organ. And I kept this in there when I got the lab from the department. It was still in there and I kept it in there because I felt it was important. You are not actually in a lab class right now. And how these labs are supposed to work, you would be getting essentially a slide. And within the slide, you would have to look through using pictures that like we're using here to identify these parts and answer these questions. And you would do it on your own. The big help is I would be there in case you got stuck or you were confused or you needed extra help to help point you in the right direction. But unfortunately, that's not the case. But remember, you can always come email me or come to any of my sessions. I will answer questions. But I wanted to keep this in here, and it seems like I'm rambling, I kind of am, but to tell you, when you are in a lab setting, always take pictures. I cannot stress this enough. The most popular class, not the most popular, but the most common class that I do teach Bio 1, I teach this Bio 2, Nutrition, Intro, A&P. But the class I teach, no matter what semester, every semester, I teach A&P 1 and A&P 2. And I cannot tell you how important it is in those classes, if you're going to move on to those classes from here, or any of the classes, micro, this course, Bio 1 is to, sorry, that's my turtle, is to always take pictures of everything. You're very lucky. I know this makes me sound like an old person, but you're very, very lucky to have the technology to utilize today that people did not have at all even when I was in undergrad. I started in 2000 at University of Houston, graduated in 2006. And, when I was going to school there, I had a flip phone. It had the worst picture capability. So what we would do is actually take an old school camera that you had to take to Walgreens or Eckerd's, or that just shows you my age, right? Or we didn't have CVS then, it was Eckerd's. And all of these things, and you would go and get them developed so that you could look at them later. Well, the great thing is you don't have to do that. And it was really expensive. You can just take it with your phone. Now, with this, is you're actually, they've taken them for you, right? It makes it hard because if you were looking under there and you were taking pictures, you would have a bit of a reference, but you get to miss one of the steps here. But I'm just telling you this because it can make you extremely successful. And what I always tell students is, especially if you're in a group, if you're in a lab by yourself, this is not going to be the case, but you are going to, in any lab that you're at, San Jack, or most of the labs, even out of for your institution, until you get, I think the only class I worked by myself was organic chemistry. Other than that, most of my classes, I at least had a partner, or I was in a group of 3 other people. And so I can't tell you how important it is to take pictures. And then if you have a group set up, you can just put that into like a Google document or something, and then have somebody else, while you're working in there, put it into a PowerPoint and actually make reviews as you go through. Now, the great thing is you have this, and I can tell you, I actually expand this lab a lot to kind of go through and give you background information, because there isn't any background information in these when I get them. So utilize some of the background information I have to make some of the questions a little bit easier. So let's go through. I just wanted to make sure to let you know that, though, because I've seen substantial increases in grades in a traditional lab setting when people start doing that. And so what you're going to do is, at the beginning, you have this procedure 1. And so this is when you're going to look at the marchanta or the liverwort and kind of check out the asexual reproduction, which you can see on the side that it's telling you what it is and what the production is that you're looking at. So what they're doing is, you can see those gemma there too. I just think liverwort is like the coolest plant ever. And you can see the gemma cups and the little gemma that's there. So they want you to sketch the preserved specimen, a marchanta plant. And so you can see that that is this guy that's right here, and you can see the little gemma cups there. And is the plant body haploid or diploid? So you can look at this. The gemma cups are on the thallus, right? So you should be able to say that is a gametophyte. And then what are gametophytes? Are they haploid or diploid? And then they're going to ask you what the plant body is called. And then they're going to ask you to draw the actual marchanta thallus below, and then label the thallus and the gemma cup itself. They're going to ask you what the function of the gemma cup is, if the gemma is haploid or diploid. So think about in terms of like spores and those things. And then you're going to look at the microscopic. And so they're showing you the gemma, the thallus, the gemma cup. And they're going to ask you to label the gemma cup and the gemma, and look at the microscopic slide, and basically compare what you see in the slide with the figure below that they give you in the lab. And then you want to be able to basically look at it, identify all the different parts, and be able to know what's happening and identify those parts. It's super cool. It's so neat. So here is just some pictures. And then at this point, they're doing table 2. So we finished the asexual reproduction. Now we're going on to sexual. And so they say, observe and draw the male and female gametophytes, a marchanta, that have been placed under the dissection microscope. And so here they are, right? And they're looking. So this is where you go and you ask yourself, can you identify the difference between male and female on here? And you should, based on what I told you. One of them have an umbrella-like, and one of them have more of what we would look at and consider to be a tree-like structure. And so they have it really nice there. If you can look and identify which is, and I can give you a hint, they show you the gemma cup on there, which is that first one that's here. Right? And that's the thallus that's on there. And then you have the archegonia here and antheridia here. And so look at these caps. And if you remember, female look like you take it out in the rain, right? And that's how you can distinguish between them. And so what they're asking you here is to take these, and you can use this one too. You can use either any of these pictures. There's several of them to give you an option. And they ask you to draw the male gametophyte. So one of these two, right, is male and the other is female. And then you should be able to label the thallus, the antheridial head, and the archegonial head and be able to tell which one is which. So, And again, that's here. These are the live, and they're just so neat looking. They're kind of creepy in some ways when they're dried out like that. And there you go, the other two. So they have 2 sets. I think that one looks good on there. That is one of the, you know, you can look at that and go, is that female or male? And here is the other that can kind of help. I think that's probably one of the better ones, though, to help you there. And so they also have pictures and drawings to help you with that. And you can also go back to some of my old ones. And then you can see the small stuff coming out that is essentially looking at the smaller ones you're going to look at are the spores. So they're going to ask you, can you differentiate between male and female gametophytes in this? And again, look at the shape of the gametic head. What type of cells will the antheridial head produce? So then you need to decide, is that male or female? Or the archegonial, is that male or female? Which of these do you think produces more reproductive cells? So think about it in terms of men and women. And the idea is that one species can essentially has to take a lot of energy to make and help generate the gamete or the zygote later, and the other doesn't. So that can help. Now, the next thing that they'll do is they go into all of the different microscopes. So at this point, you know, we're still in sexual reproduction. And if you look here, it's archegonia that it's showing you. So this is just giving you a overall holistic view and a closer view so that you can essentially see the eggs. And so in here, let's find... They're going to ask you about this, and then this one, they're looking at the antheridial head. And so this is male and sperm, right? And so you're going to look and you're going to answer, what does archegonia bear? Are they haploid or diploid? You know, can you go on that archegonia, draw it? You're going to do the same thing for your antheridial. And then you're going to talk about, and you're going to move on. Here is, you'll start seeing that this is the capsule and the zygote here and the seta that's starting to form and the placental foot and all of that. And they're going to ask you basically to identify, label. They're going to ask you what's contained in here, if this is haploid, the cells are haploid or diploid, if the sporophyte is independent of the gamete or dependent on it. And it's going to go, why would the spores be called meiospores? So look at the beginning of that word to kind of get an idea of that. And then we're going to go on to moss. And here it's a little bit easier because you only have to look at sexual reproduction. So they're going to have you draw and label the gametophytes and the sporophytes here. And they basically differentiate by both male and female. So you can see that. It's a great one of the male gametophyte right there. And so it's going through and asking you to label all these different parts. And you can use any of those that you like. And then they're going to have you essentially start observing underneath the microscope, the dissecting, the stalk, the capsules, the seta, the operculum, all of those. So you're going to look at these and get an idea of everything that you need in here. There's really nice, right? That's, you can see the spores in the capsule really nice there. And they're looking, this is what I was referencing here. And then of course your post lab. So hopefully that helps for you to identify what's going on here in this lab. And I will be continuing to make these videos for your subsequent labs over plants. I hope you enjoy this, and good luck.